All right, good afternoon. My name is Jamie Williamson. I am a professor here at Scripps Research, and I will be your host today for what is the opening lecture of our front row. This is our 8th season, and I'm kind of curious, how many people here, is this your first time at a front row in the audience? Good, we have quite a few new people, quite a few people coming back. We're pretty excited. Turns out there's about 8,400 people out there in the virtual world. We get people from all over the world getting up in the middle of the night and watching us here in San Diego. So that's great. Thank you all for tuning in. Let's see, a couple things. I want to, first of all, acknowledge we have some people here from our Science Trust and Legacy Circle groups who are supporting us. We thank you for coming, and we thank you for your support. There's one other thing that I wanted to alert you to, and that is that this is our 100th anniversary. And so there's, yeah, right. Well, it's actually 101. We didn't get the budget item approved for the extra one, but I hope you appreciate the spirit. No, it's an exciting time to reflect on everything we've accomplished, and then look forward to what's going to happen in the next 100 years. Now, okay, so pistachios. I was about 3:15. I was coming over here, and my gut and my brain engaged in a dialogue. They said, hey, I need something. Well, what would you like? Well, I don't know, what do you got? But I ended up going into my briefcase, and I pulled out some pistachios, and I had a fistful of pistachios. So this is communication along the gut-brain axis. And I think everybody can relate to what I just said, and the science of what's happening is really complicated, and it's an unfolding story. So there's two kind of ways your brain and your gut communicate. There's nerves that directly connect them, but there's also a whole set of hormones that are raging through your blood system and taking part in communication with not only the gut, but other organs. So there's this whole second language of chemicals. So we have a bunch of questions that we're going to try to address today. And I would encourage you as audience members to do some active listening. So you're going to have questions, and think about it when you're listening to the speaker. You know, think of, oh, well, I'm wondering about this, and ask yourself a question. And we will have time at the end for audience members, and hopefully time for virtual audience members to participate in a little discussion. I'll come back up here, and we'll engage with the speaker. But ask yourself a question. Sometimes your question will be answered on the next slide, but sometimes you'll hold that question to the end. It's like, aha, here's what I learned, and here's what I'd like to know. Okay. So today we're going to have Supriya Srinivasan, and she's a member of the neuroscience department, and she uses a model organism called C. elegans, which has many of the same kind of communications that we do, and it turns out to be a very effective way of addressing some fundamental problems in human health. And I'm going to let her tell you about the direct connection, but it involves hormones, and she's setting the stage. Now, I mean, I almost got up here and sang a few jingles that you might have heard on TV about certain kinds of drugs that are on the market. I'm sure you've heard most of them. But that's the research that Supriya is doing now. She's doing the research that's going to lead to the next. But really, there are new pathways that are going to be discovered and new medicines that will come out of the things that she's working on. So I'd like to invite Supriya up to the stage, and I will return at the conclusion of her talk. Welcome, and please join me in welcoming her.
Thank you. That's kind of setup. Thank you, Jamie, for that incredible setup, and thank you all for turning out in person. It's always so special to have a live audience. Your patronage, interest in science, and support are truly intrinsically valuable. Also, a big thank you to the virtual audience that I know is all around the world, so thank you so much for making the time. So our topic today is the gut-brain axis, or the bidirectional communication between the brain and the gut. And my goal is to share with you not just what we know, but how the process of knowing, or the process of scientific discovery, occurs, and what the fascinating biology is telling us about metabolism and how it even plays a role in longevity. So all the organs in our body are actually in a state of constant communication, as Jamie mentioned. So a helpful analogy is more than a dialogue, like a symphony in which every instrument has a role to play that together orchestrates a beautiful piece of music. And so it is with our bodies. Different organs are constantly communicating with each other through what are known as hormones, when the communication goes from one organ to another, but can also happen through different cell types using what are, for example, neurotransmitters in our brain and in our peripheral nervous system. And the way in which that occurs is that there's one organ or one system that's relaying the information or the hormone, and then the receiving organ has what are called receptors, or proteins that receive the information and act accordingly in proportion to what the original organ has relayed. And so this mechanism of organ communication sort of together stays perfectly in tune to generate a state of internal homeostasis where the body is humming along nicely. However, a classic hallmark of disruption in this internal homeostasis occurs when any kind of metabolic dysregulation occurs. And so by studying metabolic dysregulation, we can get at the root of how different organ systems have gone awry. And in fact, metabolic dysregulation is a root cause underlying a lot of diseases that affect us today, including, for example, diabetes, muscle loss, heart disease, and stroke, and even more complex diseases like neurodegenerative disease, certain types of cancers, and so forth. And so my scientific origins in this sort of general problem of how a signal that's sent from one organ is received by another began when I was working on the melanocortin-4 receptor, which is a receptor in which mutations are associated with the most common form of monogenic obesity in people. And so I was trying to understand during my postdoctoral studies how mutations in these receptors can lead to the enormous physiological consequences that we see. But this work was being done in the backdrop of a genomic revolution in the early 2000s, in which the genomes of many different species, including the model systems that people were using in labs to study fundamental biological processes, were unfolding, right? And one of the biggest surprises that came out from the genomic revolution was with the sequencing of the human genome in 2001, that there was incredible shared genetic ancestry and shared genes with common functions across these different species. One model system we're going to talk a lot about today, the humble C. elegans, depicted here, has somewhere in the range of about 70% of genes that are common between C. elegans and us humans. And so what that allowed us to do was to study not just one gene at a time in humans, sort of hoping to get at discovery, but rather uncovering new aspects of any biology of interest. So for example, the gut-brain axis at speed and scale. So this is a picture or a video of C. elegans. It's a tiny microscopic nematode. It's about 1 millimeter long, so of course this is a view through a microscope. As I mentioned, about 70% of the genome is conserved with humans. We have incredibly powerful genetic and molecular tools that allow us to take out individual genes and put them back in any cell of interest. The life cycle is very short, it's about 3 days, and the lifespan is about a month. So what that means is that we can do a lot of studies at scale and speed in living animals. So shown here on the right panel is a little 24-well plate, so it's about sort of this big, and each well is about 1 centimeter across in diameter, and we can put 30 or 40 animals in there and screen at scale and speed across the entire genome for any phenotype of interest. So what are some of the things that we can measure? On the gut side, the anatomy is incredibly well specified and well known. There are exactly 20 cells in the intestine, and their functions are very similar to those of our intestine. They are chock full of lipid droplets, and what you see here is the germline. So this female is about ready to lay her eggs. The biology happens incredibly quickly, right? So the animal only lives for a month. If you take an animal off food for about 3 hours, more than 80% of the fat is gone from the intestine, so it mobilizes fat as a source of energy. The body of the animal is transparent, and so what that means is if you develop fluorescent reporters to study any process of choice, you can measure that in living animals. And so we've developed, over the years, multiple different reporters. We're just showing 2 here. One reports an animal in the fasted state, and the other reports an animal when it's been put back on food. Okay, what about the neuronal side? We know exactly how many neurons there are, and through genetic methods, we can put in fluorescent tags, singly or in combination, in every single neuron, allowing us to mark both in space and time what these neurons, where these neurons are located, and by putting in fluorescent reporters that mark the activity, we can actually measure the activation state of these neurons. We can also do things like measure peptides or hormones that are secreted from one tissue to another by developing assays that allow us to measure what is found in the circulation itself. Okay, so that brings us to the focal points that I wanted to hit in today's lecture. I'll start with sort of at the beginning, which is our tryst with serotonin, as I always like to call it. Serotonin is a principal driver of fat loss in many different species. It's made from the amino acid tryptophan, and an enzyme called tryptophan hydroxylase is the rate-limiting step for the conversion of tryptophan to serotonin. In C. elegans, the enzyme that makes serotonin is found only in 2 pairs of neurons in the head of the animal. And so it works something like this, right? The neuron that makes serotonin releases it into the synaptic space between 2 different neurons, and there's something called a receptor, which receives the serotonin signal and effects a response in the downstream neuron. What we found was that if you take away the enzyme that makes serotonin in neurons, it leads to a massive accumulation of fat in the intestine, as a classic example of something that's altered up in the nervous system, but it's having its effect in the gut. On the other hand, if you take away this selective serotonin reuptake transporter, which will be familiar to some of you because it's the target for Prozac, if you take away the reuptake transporter resulting in more serotonin in the synapse, then these animals become pretty lean. So this sort of general concept of putting genes in and out of different cells in different locations can be used to identify the neural circuit that drives serotonin-mediated fat loss in the intestine, using genetic tests that are sometimes called necessity and sufficiency tests. So are these genes necessary and sufficient to manifest a certain effect, that is to say, fat in the gut? So we worked out the neural circuit. We also went looking in the intestine for how these effects might be unfolding. And it turns out that the way serotonin works is by triggering an enzyme whose job it is to convert stored fat to usable energy. So what this enzyme does is it literally converts or breaks down stored fat via the beta oxidation pathway in the mitochondria that generates energy. But all of this, I mean, this was good, but it just led to a big question in our minds because the gut in C. elegans is not directly innervated. So if you have all of these effects in the nervous system manifesting in the intestine, how does that work? To answer that question, we embarked on something called a genetic screen, which is simply a way in which you can remove one gene at a time from the entire genome and ask, does the presence of this gene have a role to play in what I'm measuring, which is the ability of serotonin to affect fat loss. And so by screening the genome, we found a single hit, which ended up being a secreted peptide called the tachykinin peptide. We also found the receptor for this peptide, and the name of the receptor is not too important, but just know that it's a tachykinin peptide that acts as the receiving signal for the relaying tachykinin signal. Okay. In data that I don't have time to show you, many other people in the lab were sort of simultaneously trying to figure out how other neural inputs regulate fat stores. And what they discovered was that the tachykinin brain-to-gut pathway is the final common pathway by which many different neural signals are relayed to the intestine to regulate metabolism. Okay, so we wanted to understand a little bit more how this process of converting fat to energy was working. And one of the things we noticed was that if you give these animals a little bit more tachykinin peptide than what they normally have, it really boosts what's called their oxygen consumption rate, which is simply a way of measuring their energy expenditure. And this is a theme we'll be coming back to again. So it turns out that if you boost tachykinin signaling, it increases energy expenditure. And we became interested in what else might be going on, because increasing energy expenditure chronically might have other effects. And this is what we discovered. We found that in the intestine, during the process of fat oxidation and mitochondrial respiration, there's a mild mitochondrial stress response that's evoked. And through the actions of these genes that confer between the mitochondria and the nucleus where the genome sits, there's a protective response that's evoked. And the reason we know that this is a very important part of the biology of the animal is that if you remove the key protective response in the gut, it brings down the lifespan of the animals. And so what that's telling us is that as long as this process is allowed to unfold in an uninterrupted way, the stimulation of the tachykinin signal to drive fat loss has no deleterious effect on longevity. But while we were working on this tachykinin pathway, we had a hunch. So when you have a brain-to-gut signal, you start wondering, well, how does the nervous system actually know to send this information? Does it receive any kind of feedback cues from the gut? And so we also had some circumstantial sort of evidence that if you deplete the fat stores in the intestine, it changed the properties of the nervous system, not just of the tachykinin signal, but of some other neurons as well. And so to ask the question more systematically of what a gut-to-brain signal might look like, we embarked on another screen. In this instance, the genetic screen was not to measure fat stores, because this time we are going from the gut back up to the nervous system. And so we change individual genes of the gut and ask, to what extent does changing those genes alter the tachykinin signal back in the nervous system, right? So the screen looks something like this. We can now automate them. We don't have to do them by hand anymore. We can get computers and annotation software to give us answers, so we can do these screens very quickly. And the answer that we got was really interesting. The most profound hit from the screen was an insulin peptide. We all know insulin, right? It's the hormone that's normally secreted in response to incoming food, right? So it's what we would call a postprandial signal. You eat something, your pancreas produces insulin, the glucose and carbohydrates that are produced as a result of digestion are disposed to the different muscles of the body and different regions of the body. But that's not the kind of insulin we found. We found sort of the opposite. It's something called an insulin antagonist. a molecule that is very different phylogenetically from canonical insulin, which is all sort of down here in humans, IGF-1, IGF-2, and insulin. But the insulin that we found was phylogenetically completely different. And to our surprise, secreted in the fasted state, right? So it's a gut-secreted insulin that is, you take the animals off food, the secretion goes up. You put the animals back on food, it goes down. You take them off food, it goes up again. And so this is a gut-secreted insulin antagonist that's released primarily in the fasted state and modifies properties of the nervous system, including the tachykinin peptide that we had found. Okay, so it's a secreted insulin, and we, of course, became curious on the question of what happens if you remove this insulin. And interestingly, we found that this fasting secreted molecule, when it's genetically deleted, when it's removed, has a pretty dramatic effect on lifespan. That is to say, loss of IN7 shortens lifespan, suggesting that this gut-secreted molecule during fasting may have some really important role to play in maintaining the longevity of these animals. So fasting drives a signal, but refeeding brings it back down, right? So we wanted to understand a little bit more about how this process is actually working. And so we don't have the answers to this yet, but we're taking sort of a two-pronged approach, right? So first of all, just like our intestines, the worm intestine has a lot of bacteria, which is a major food source for C. elegans. And so one question we want to ask is not just which bacterial species are represented, because in the lab at least, the animals are given a very controlled diet, right? So we know exactly what bacteria they're getting. But what we want to know is what specific signals from the bacteria are being sensed by these gut cells that are allowing the insulin to be either secreted into the circulation or not. And so for that, the approach, we're taking two different approaches. One of them is to give the animals a bacterial genomic library in which one gene at a time has been deleted. So instead of working on the host genetics, we're going to work on the bacterial genetics and take out one gene at a time and say, and ask, the absence of which gene results in a response that the animal now can't see, right? Does that make sense? So in other words, we want to know which gene is required for an animal to go from its fasted state back into its refed state. And by doing that, we're hoping to pull out specific bacterial signals that the host is responding to that we could potentially manipulate for regulating insulin secretion, as well as for understanding how this process actually works. The other approach that we're taking is on the host side, right? So we can generate, using spatial and other methods, transcriptomes, or the readout of the genome from different parts of the cell in different states, right? So using a combination of genetics and environmental disruptions, we can already see that different parts of the intestine respond very differently to food deprivation and restoration. And so by using a combination of host and bacterial genetics, we're hoping to be able to get at the sort of questions around what specifically the gut is seeing and in what way it's relaying information and what impact that information has to the rest of the animal. So while we're still sort of figuring out this communication between the nervous system and the gut, I want to focus a little bit now on how these sorts of findings, I mean, you really should be asking yourself, like, the worms are really cool, but what does this mean for me, right? So, and that's exactly what we asked ourselves, and started thinking about how we can translate some of these findings from basic science towards clinical applications. And here the story begins with this tachykinin receptor, right? In worms, it's called NPR22, but it has astonishing conservation sequence-wise with what we see in humans and in other model systems, including mice, which are really a workhorse for a lot of the work that we do. Okay, so in worms, it's expressed in the intestine. There's some variation in the intensity of expression, but it's pretty well expressed across the intestine. The single-cell sequencing efforts of different tissues within the human body can be leveraged to figure out where your favorite gene might be expressed. And so when we went looking for our gene, which in humans is called TACR2, we see very interesting expression specifically and selectively in the intestine. And it's thought to play a role in digestion, although this has not been functionally validated. So we went looking in the mouse intestines. So what you're looking at here in green is antibody staining of the mouse tachykinin receptor. And what you're looking at here in blue are the cells of the intestinal tract. So you're seeing here sort of the villus that protrudes into the lumen with the musculature lying underneath. And so the tachykinin 2 receptor is really pretty robustly expressed, particularly in the anterior half of the intestine, along the enterocytes, or the cells that are responsible for digestion and absorption of nutrients. So after doing some sort of preliminary experiments to see whether the way we were thinking about this was correct, we decided to go ahead and use CRISPR-mediated tools to delete this gene from the mouse genome. So what you're looking at here are up top wild-type male and female mice. Actually, you're looking at the intestines of those mice that have been given a high-fat diet for a few weeks. And down here, you're looking at what happens when the receptor has been knocked out from the intestines. And what you see in black here is the fat that's been stained. And so what you see is when you take the receptor out, you get this massive accumulation of fat in the intestine. So if you knock the receptor out and it builds up fat in the intestine, it's logical to think that if you stimulate the receptor, maybe it would do the opposite. And that's exactly what we went looking for. So we went looking in the literature to see if there are small molecule modulators or any kind of modulators that can be used to tweak the receptor, to modify its functions, turn it on, turn it off, and so on. And we found some tools. They're not amazing, but we did find some tools that we could use for experimentation. And in collaboration with Caliber, which some of you may know is the drug discovery and translational arm of Scripps Research, we embarked on kind of a simple experiment in the end, actually, in which we gave mice a high-fat, high-sugar diet until they really gained a lot of weight. So lean controls are down here. They gained quite a lot of weight, so this is like a really heavy mouse, and gave every other day injections of a compound that is known to stimulate the receptor. In other words, it turns the receptor on. And both at the low dose and at the high dose, we observed a pretty dramatic drop in body mass. These mice that have been given this high-fat diet are also diabetic, as you can see by their high levels of blood glucose, which this compound pretty effectively normalizes. The mechanism of how this is working is still a huge open question, but sort of simply measuring phenotypes, including measures of food intake. So we see an initial drop in food intake that seems to normalize. For the aficionados in the audience, if you are wondering about lean mass versus fat mass, we did a host of measurements, only some of which I can show here, but the change in body mass we see is coming primarily from a drop in fat mass, with lean mass being relatively preserved, suggesting that this may be a very effective mechanism to elicit fat loss. So, maybe you're convinced, maybe you're not convinced, but what should really convince you is that last November, there was a paper from a group funded by Novo Nordisk and in collaboration with Novo that showed that the NK2R receptor can be used, again, to leverage an increase in energy expenditure to treat metabolic diseases. And so in a way, we come full circle. We are planning to leverage the human NK2R receptor to find small molecule modulators for weight loss, type 2 diabetes, and cardiometabolic benefit. But you don't undertake something like this alone. We are working with two mighty powerhouses. The tremendous intellects of Hugh Rosen and the Rosen Lab and Ed Roberts and the Roberts Lab have already led to successful medications that are in the clinic today. You may have heard of ozanimod, which was approved in 2020 for multiple sclerosis, and then fast-tracked for ulcerative colitis in 2021. So our labs are partnering to make what we think will be very effective small molecule therapeutics for weight loss, type 2 diabetes, and broader cardiometabolic benefit. So I hope that I went some way towards convincing you that a tiny nematode can tell you a lot about biology and medicine. I couldn't go too deeply into anything, but I'd be happy to address more questions that you might have about the broad roles of serotonin, tachykinin, insulin antagonism, and how we might take findings like this that come really from basic science that graduate students and postdoctoral fellows work on towards what will hopefully be tremendous therapeutic benefit. So I want to leave you with the sort of broader vision of what we're trying to achieve in the lab. We think that the gut-brain axis is rich with hidden signals. So on the one hand, we know that through efforts like the ones that I've described, as well as using whole brain GCAMP studies, we can uncover a rich plethora of neuropeptides that are functioning both within the nervous system and across different organ systems to regulate physiology, behavior, and metabolism. Using spatial transcriptomics and other methods, we are uncovering receptors for those signals, as well as additional peptides that are being relayed to the rest of the body. And so we think that this broader sort of way of thinking about how organs communicate from one another will lead to a subset of molecules for which we can develop functional assays and screening for discovery efforts to ameliorate or circumvent some of those metabolic diseases I alluded to at the beginning of the talk. With that, I will acknowledge the key people that did the work. My lab members are listed here. Everybody in asterisks is here in the audience, and I believe have prepared something for you outside that you could take a look at. I've had many collaborators along the way that have helped us with many different aspects of the work. Scripps Research is really an incredible place to work, I have to say. I mean, no bias there. But some of the work that I've talked about, and very importantly, some of the thinking that has shaped me and shaped the work in my lab to be able to take these very basic, fundamental, unknown questions and take them in a direction that may one day lead to therapies, it's a very Scripps sort of thing. And in conjunction with many of the faculty I'm acknowledging here, as well as some of the key people at Caliber who really encouraged me to start thinking in a translational way, I could easily say that I'm not the same scientist I was when I came here. The C. elegans community is very interactive, very collaborative, and we freely exchange information without MTAs and so on. And that's really very unusual in the scientific community. So big shout out to the world of C. elegans. With that, I will thank you and take any questions.
Okay, thanks, Supriya. Why don't you come over and have a seat? So for those of you in the virtual audience, I have an iPad by which I can receive your questions if you type into the chat. I'm not sure I'll get to 8,000, but we'll give it a shot. And then if you're in the audience, we have Jedis armed with lightsabers that will indicate that I should call on you with a question. So maybe while you're getting your nerve up to ask a question, maybe I'll ask one. And
Sure.
so I was particularly interested in the fat accumulation when you knock out tachykinin, and then if you inhibit it, then you lead to weight loss. But
No, wait.
sorry,
If you activate it, you get weight loss.
sorry. So
If you inhibit,
yeah. So okay, fact check. So I'm kind of curious about, is there crosstalk? Is there anything else going on? Because you described at the beginning, there's lots of hormones, and you have these compounds that sort of do something. But is there any crosstalk with any of the other systems that are involved in weight loss and metabolism?
That's a really good question. So I'll answer it in two ways. First is, I made it out like there's one tachykinin and one receptor, but actually the tachykinin receptor that I talked about is part of a family of 3. So there are 2 other receptors that are also found in the intestine, but at much lower levels. The endogenous ligand for NK2R, so the peptide that's normally made in the body, actually does have crosstalk with the other receptors, which is why it would make a terrible therapeutic. So achieving selectivity for any one of these receptors, whether it's tachykinin or anything else, is going to be a crucial thing that we have to hit. The other thing, of course, is where else is tachykinin expressed and what other hormone systems is it speaking with? Those are big unknowns that we're trying to address. The data so far does seem to suggest that it's circumventing, in other words, sort of bypassing the canonical leptin pathway, which is it's actually a very important hormone that functions from adipose tissue to the brain and is thought to play a role in maintaining long-term energy balance, not moment-to-moment, like day-to-day variation in food intake. It's thought to bypass leptin, but the mechanism right now is totally open.
Okay, great. So do we have a question from the audience here? There's one up in the back.
Hi, wonderful talk. So actually just riffing off of the comment with the natural ligand interacting with the receptor, can you maybe talk a bit about why, the decision on the modality for a small molecule? Obviously, Scripps is world-class in that approach, but there's obviously many choices in your repertoire now that you've discovered this new signaling pathway to prosecute the target. So can you talk a bit about your decision there with small molecules?
Yeah, absolutely. That's a very astute question. And of course, we and others have considered peptide therapeutics, like the ones that we are all familiar with, AKA Ozempic. And I know that people are working on combination therapies and so on, but I think our thinking is trying to look a little bit ahead and considering a world in which people might not want to take daily injections and instead want to go in pill form. So that's one of the primary reasons we're thinking about that. The matter of achieving selectivity is another important consideration.
So I'm going to aggregate some questions from the virtual audience into kind of a little package. But basically, people are interested in the possibility of additional weight loss medications, but what's the time frame? I mean, we're getting heavier here, but seriously, I mean, I looked up the arc of from when Ozempic was discovered to when it became clinically applied. But but I think it's helpful for people to just understand. You've got this idea, and so where does it go, and how long is that what's that look like?
Well, I see this is a good question, right? So GLP-1 the incretin effect discovered in the 1970s, right? And by the time it reached the clinic as Ozempic, I think 2017, right? So we're talking, we don't have that much time. There are really, the science is the science. There's a lot of screening that has to happen, and many of you may have heard, there's many steps in the drug discovery process at which a molecule that looks very rational and beautiful might fail. So there is an intrinsic process that is designed for people's safety that we cannot circumvent. So there's that timeline, and then of course there's the matter of resources, and whether in a, I would say, relatively crowded space, there would be, no pun intended, appetite for another molecule. So timeline-wise, I mean, the number that's generally thrown around, although I won't be held to it, is about 10 years. We sure hope that it's less.
Yeah, I mean, there's plenty of advances in drug screening and genomics that are going to greatly accelerate the GLP-1 inhibitor timeline. So that's the good news. But it's going to be a long road. Okay, a question up here.
Thank you for making the talk easy for a layperson to understand. So can you comment on why different SSRIs like Prozac have different effects on weight gain and weight loss in a patient?
So I wasn't medically trained. I'm a basic scientist, so I'm not qualified to be able to make any sort of statements about how medicines work in people. I will say, though, that the phenotypes that I described for SSRIs have been well documented acutely, right? What happens chronically, though, I mean, humans, we share a lot in genes with worms, but we're people, right? I mean, we're fundamentally very, very different and much more complex. And so the compensations that happen, both as a combination of genetic composition, right? It's also not individual genes. It's constellations of genes with subtle variations working together that make us both similar and different from one another. So it's a little hand-wavy, but it's most likely a combination of genetic variation and environment that lead to those differences.
So I have a question that kind of interested me. So C. elegans eats bacteria, and typically you feed it good old E. coli, right? Right. And so then now we're looking at mice and people. That's not really our food, but our gut is just filled with hundreds of different kinds of bacteria that populated at steady state, and we all have a different gut microbiome. So is there any crosstalk between our microbiome and the function of these receptors?
That's a good question. So Anthony in my lab, who worked on the NK2R project, was interested in the same thing and did some gut microbiota analysis that I didn't think I'd have time to show, but I can tell you. So the experiment was...
This wasn't a plant, by the way. This is
right. This is not at all.
This was real. This is real.
Right.
Okay.
So the experiment is very straightforward. Normal animals that are lacking the receptor, that have all been put on either a regular diet or a high-fat diet for 1 week, right? So really short amount of time, not enough to really change the metabolism of the mice, but enough to change what's happening in the gut. And took the microbiota and had them sequenced. And one of the first things that came up was something that's already been well described, which is there's a ratio between these two different bacterial clades called firmicutes and bacteroides, and the ratio of those will tell you if the diet has been either a normal diet or a high-fat, high-sugar diet. And in fact, the ratio was very predictive in wild-type animals, just the way we'd expect. But in the mutants, so the ones lacking the receptor, they didn't show the same profile. They actually showed a more protective profile. So even though they're accumulating fat in the intestine, the microbiota looked different. Whether, though, the difference in the microbiota comes from the actual absence of that gene or from the underlying mechanisms that make the intestine store fat, we don't know yet, right? So that's sort of a complex chicken-egg thing for us to tease apart, but we do see differences.
Okay, terrific. So I saw a Jedi here.
Thank you for the nice talk. You mentioned at the beginning that the mutations in the melanocyte 4 receptor is a monocausative driver of obesity. How prevalent is that in the population? And can you study it in C. elegans? And how many different genetic causes are there of obesity, and can you study each of those in your system?
Okay, that's a big question with 3 sub-questions. Let me try and get at them. So the mutations in the melanocortin 4 receptor that lead to obesity are very, very rare. Actually, they're very extreme, and they run in families. So it's not the quote-unquote garden variety metabolic dysfunction that you would see. They're quite rare. Less than 0.01% of the population. And as I mentioned, they don't really arise de novo, right? They are inherited within families. So I did have, when I first started working with C. elegans, I did have the idea of putting the human melanocortin 4 receptor into worms and looking to see how it worked. But after thinking about it a little bit more, kind of realized that the leptin-melanocortin axis, as it's called, has arisen as a result of vertebrate evolution. So invertebrates don't actually have that whole axis. So theoretically, it is possible to put a receptor in and study it. And actually, we've done that for other receptors and toxins and that sort of thing. But the question really became, even if you did get the receptor to express, how much would the biology that you uncover be a faithful representation of either the worm or the human? And without having a good answer to that question, I didn't proceed. But your other question about, are there other forms of genetic obesity? There are. So the leptin receptor itself and the production of the hormone leptin itself was identified in part through human genetic studies. So there's a rich biological history of identifying human genes that run, that are causative for disease that run in families, and studying them in model systems. And that has and is being done. In fact, the neurokinin 2 receptor also has mutations in it that are associated with obesity, again, very rare.
Okay, I think we want to maybe wrap up, but I'll just ask you just finally kind of this, I'll give you a softball. What are you most excited about doing next?
Wow, there's so much to do.
Well,
I think that if we can collaborate with some of the leading scientists at Scripps and Caliber and really push the envelope on whether a basic science project can be taken to the clinic for therapeutic benefit, I think that would be tremendous.
Yep. Agreed. That's great. Okay, we're gonna pause here. I want to thank everybody on the virtual audience for tuning in. Thanks for joining us. For those of you here in the audience, I'm going to give your brain a message, which is that your gut will receive nutrition outside if you join us for a little bit of a reception. And as Supriya mentioned, her lab people are here. They have tags, I think, and then there is a C. elegans display. You can go view them in the microscope. And so the last thing I have to do before you all run off is the next one is, it's March 19th, the next front row, and it's Andrew Ward, and he's going to be talking about the tremendous story about finding the COVID vaccine. Some of you might be familiar with it, but he's a tremendous scientist, and you should really tune into this. And if you come here, you'll find me in the front row.
Thank you.